Dolly, a clone who has lived a life of awe and infamy. Also known as 6LL3, Dolly is a clone of a black-faced ewe created in July 5, 1996 at the Roslin Institute in Edinburgh, Scotland by embryologist Ian Wilmot and his team of other scientists. She was created by the process of nuclear transfer, which is done by transplanting a nucleus from a cell such as the mammary gland of a Finn Dorset sheep into the enucleated egg of a Scottish black-faced ewe. The nucleus-egg combination was stimulated with electricity to fuse the two and to stimulate cell division. The new cell divided and was placed in the uterus of a black-faced ewe to develop. Dolly was born months later. She was named after the American singer Dolly Parton due to Dolly's original cell being taken from her mother's mammary gland. Dolly began to live in a sheltered environment at the Roslin Institute, which later gave her crippling arthritis and then lung disease. She was put down at the age of 6 on February 14, 2003, which is half the age of normal sheep. When Dolly was presented to the world changed its views of cloning and what possibilities are there. No longer being science fiction, being able to restore extinct species such as mammoths, cure diseases that haven't been able to before, but the one thing that people feared out of all the possibilities was the idea that humans had the possibility to be cloned as well. Dr. Wilmot, though, described the process of cloning another human as illegal and disgusting. The rest of the world, though agreed, wasn't going to take his word. Animal rights activists were offended to know the effectiveness of cloning, and even the US President Bill Clinton sent a special task force to see into the Roslin Institute and to see if their research was ethical. One of the ethical issues that people have is that even a successful clone is not exactly like the original. This was assumed by Dolly having arthritis and being put down due to her suffering from lung disease at half the age of average sheep. Dolly's health issues are derived from being in captivity at the Roslin Institute, and this was proven when researcher Kevin Sinclair had made 3 sister clones from the same sheep that Dolly was cloned from and raised the sisters on an actual field. All 3 of them lived to be the ripe age of 10. Even though Dolly was the one who created mass hysteria around the world, credit needs to be given where credit is due. Ian Wilmot, the embryologist and head scientist in the project that created Dolly, was born in Hampton, Lucy, England, near Warwick, was introduced and later loved embryology when he was an undergraduate at the University of Nottingham. Wilmot was mentored by G. Eric Lamming, a world-famous expert in the science of reproduction. From then on, Wilmot said he knew animal genetic engineering was his life's quest. In 1971, he went on to Darwin College at Cambridge, where he received his doctoral degree 2 years later after submitting a thesis on freezing boar semen. He headed almost immediately to Scotland, where he joined the Animal Breeding Research Station, an independent animal research institution financed by government and private donors that eventually became the Roslin Institute in Roslin, Scotland. After the creation of Dolly, Wilmot discussed within an interview the ethics of cloning and where he stands on certain issues. We didn't set out to clone adult cells. We set out to work with, ideally, embryonic stem cells, or things like that. Ian Wilmot openly said that Dolly was a happy accident, but does say it was an accomplishment, that it was an essential breakthrough into programming adult cells. The main goal that Wilmot was trying to achieve was not creating duplicates of animals, but for creating a cure for ALS, and the closest that he has gone is the discovery of induced pluripotent stem cells, or also known as iPS cells, that in theory could be used to revolutionize the medical field. After more research was put into cloning, people began to see how much potential it had. One of the definite pros of cloning was its viability. Cloning could not only help reproduce dying species, it can also help other species more than just balancing the food chain. With the use of cloning, certain vital organs like hearts and livers can be duplicated and be used as a replacement in transplants. Another use is that they can be used to produce certain proteins that can possibly be the cure for certain hereditary diseases like diabetes and heart disease. A very crucial use of cloning is that the stem cells from the embryos can help restore degenerative mental diseases like Alzheimer's disease. As surprising as it may seem, Dolly isn't the only clone. She was the first mammalian clone made by somatic cell transfer and from cells in a laboratory. Clones before Dolly were tests to figure out how the embryo in different animals work and how to manipulate their process. When cloning as a whole began to develop, it involved less complex animals like frogs, and after Dolly there were more advancements in embryonic studies, such as the first primate to be created with embryonic cell transfer with cells in a laboratory by Li Meng, John Ellie, Richard Stouffer, and Don Wolfe in 1997, to human embryonic cells created by somatic cell transfer by Shokrat Metalopov and colleagues in 2013. After Dolly's legacy came to an end in 2003, being put down after suffering a lung disease, the focus of cloning has divided to either accepting it or not. Only a few countries even allow cloning, such as South Korea, where a biotech lab named Suam is willing to clone your pet for $100,000 using the same technique as Wilmot used for Dolly. Other countries use cloning for agricultural purposes, such as the US and China, to capitalize on the genes of a few extraordinary specimens. But the European Parliament voted in 2015 to ban cloning animals for food. Yet other scientists like Wilmot utilized knowledge of embryonic programming, which led them to an access of induced pluripotent cells, or iPS cells, artificially made stem cells that can be programmed to be in a state similar to infant cells, which can mature into a variety of specialized cells, such as nerve cells. Even though that they were speculated in Wilmot's time, stem cell biologist Shinya Yamanaka was inspired by the creation of Dolly and saw the possibilities of nuclear programming and began to test and create iPS cells, which has now become the basis for most of modern-day stem cell research.